So first I would like to thank, of course, the organizer for this very kind invitation to present here our work about the endocannabinoid system, which is a link between mitochondria and brain function, and including memory. So, of course, I'm not going to teach you about mitochondria. I guess that in this room everybody knows what is this organelle. But maybe I can tell you a little bit about this endocannabinoid system, which is the plant. Cannabis sativa is known for 5,000 years now, but the active compound, tetrahydrocannabinol, or THC, was discovered only in 1962. The endogenous receptor of this compound, the CB1 receptor, was found 30 years later, very close to the identification of anandamide, which is the endogenous ligand of this receptor. So what this endocannabinoid system is doing in the body, in the brain first, this system is involved in many functions, such as fear, locomotion, or food intake. But the system is also involved in other organs, such as muscle, heart, and liver, regulating the circulation or the reproduction. So, how does it work, this system? It's quite easy. You have an action potential leading to the release of neurotransmitter. This neurotransmitter going to induce an increase of calcium in the postsynaptic neurons, and then leads to the synthesis and the release of endocannabinoids. These cannabinoids then are reaching the receptor at the presynaptic neurons, and then this receptor regulates the action potential, decreasing the neurotransmitter release. In the cell, CB1, THC, and anandamide can activate CB1, which recruits then the GI and GO proteins, and which regulates adenylyl cyclase and PKA pathway. This system can also regulate the MAP kinase pathway. So why should we consider the endocannabinoid system when we are working on mitochondria? First, because soon after the identification of THC, a lot of papers came out showing effect of THC on mitochondria. They attribute this effect to the lipidic nature of the THC. But what is very important is first that this paper came out long time before the identification of the receptor. So now that we know that there is a receptor for this compound, could we have some specific effect? First, I would like to stress that there is cannabinoid in the mitochondria. For example, anandamide or 2-AG are present in the organelle. About 1/3 of CB1 ligands are present in the mitochondria. Then we identify with Giovanni Be that the main receptor, CB1, is also present in the organelle. Indeed, when I joined Giovanni's lab, we performed this immunogold staining against CB1 on brain section from wild type and CB1 knockout mouse. And we found that in mitochondria, you have some labeling of CB1, which is not present in the CB1 knockout. Of course, we confirmed this result by Western blot after isolation of mitochondria. So there is the the ligand and the receptor. Now, the next step was to identify any effect on mitochondrial function. And we measure for this the mitochondrial respiration, and we found that activation of CB1 inhibits mitochondrial respiration. For this, we measure the complex 1 dependent respiration on mitochondria isolated from wild type and CB1 knockout mouse. And we measure the respiration. We had some different concentration of cannabinoid, WIN or THC, and we measure the respiration. And here you can see in the wild type mitochondria, you have decrease of the mitochondrial respiration, which is not present in the CB1 knockout. Same result with the THC. So it is true that the activation of CB1, according to this result, regulates mitochondrial respiration. But how this empty CB1 signaling pathway can regulate the OXPHOS? This was our next step. And of course, because CB1 is involved in adenylate cyclase and cyclic AMP signaling, we check the phosphorylation status of mitochondrial protein. And here on mitochondria isolated from brain, we had THC, and we found that under THC treatment, you have modification of mitochondrial phosphorylation profile. Here, for example, you see that this spot is disappearing. How MTCB1 can regulate this phosphorylation? That was the main difficulty we had. So we think that the MTCB1 is on the mitochondrial outer membrane. So how this receptor can regulate the OXPHOS? We postulate that this regulation go through the SAC, the soluble adenylate cyclase, and the PKA pathway. To confirm this hypothesis, we measure mitochondrial respiration, which is inhibited by THC, under the presence of different chemicals. Here, for example, we use pertusis toxin, which is an inhibitor of the G-alpha protein. And you see that if we had THC, we have a decrease of mitochondrial respiration, inhibition of mitochondrial respiration. But when we block this pathway by adding this compound, you impair the THC-dependent inhibition of mitochondrial respiration. We did the same thing using different drugs, like bicarbonate, which is an activator of the SAC, and cyclic AMP, which is activator of PKA. And in both cases, you impair the THC-dependent inhibition of mitochondrial respiration. So then we check how this can directly affect the phosphorylation status of OXPHOS protein. And especially, we focus on the NWFS2, which is a protein of the complex 1. And in mitochondria treated with THC here, you have a decrease of the phosphorylation of NWFS2 here. And this is not the case in knockout animals, knockout mitochondria. So what the main things, the main interest was to identify if this MTCB1 signaling leads to any physiological impact, and especially any impact on brain function. To study this, we use a genetic approach, which was based on the fact that we found, using in silico approach, that MTCB1 have 40% probability to go to mitochondria. But when we remove the 22 first amino acid of this protein, we decrease this probability to less than 1%. So we thought that removing this 22 amino acid will be a good way to discriminate between plasma membrane CB1 and mitochondrial CB1. So we, of course, verify this in vivo. For this, we use stereotaxic injection of adenovirus in hippocampus of CB1 knockout. We inject either the full CB1 knockout or the delta N22 CB1 in the hippocampus. And we verify by immunogold staining the localization of both constructs. When the CB1, the full CB1 knockout is expressed, you have staining in mitochondria here and also in the plasma membrane. But when you express only the delta N22, you have only labeling on the outer on the plasma membrane and not in the mitochondria. So delta N22 is expressed only on plasma membrane. And we verify that both that removing this 22 amino acid that did not affect the ability of CB1 to bind its compound. We perform GTP gamma binding, and there is no difference between both receptors. We verify whether this delta N22 affect or not the mitochondrial respiration. Using MEF knockout, we express either MHA or CB1 or the delta N22. And we had a cannabinoid, HU 2010, 210, to inhibit mitochondrial respiration. And we found that when we express CB1, we have decrease of mitochondrial respiration, which is not present when we have expressed the delta N22. We also check whether this delta N22 is functional by measuring the activation of cytosolic pathway, the phosphorylated pathway, and we found that there is no difference between expression of CB1 and delta N22 on the activation of the ERK pathway. So then we analyze whether this delta N22 can have a different effect on brain function. For this, we use this behavior test, which is the novel object recognition test, which is dependent of the endocannabinoid system. The aim is we put a mouse in a maze and the two objects here, the yellow, and this mouse will spend some time to visit both objects. And the day of the test, we put a new object here, the green triangle, and the mouse will spend again some time to visit this object. And we calculate memory performance. And we perform this assay using injection of adenovirus, either expressing the full CB1 or the delta N22. And here you see when we measure the memory performance on Y type, when we put the cannabinoid, you have decrease of the memory performance. This is not present when we use the knockout. When we put express back in the hippocampus, we express back the CB1 receptor, you have this decrease, sensitivity to the cannabinoid. And when we express the delta N22, you don't have this decrease. So this means that the regulation of mitochondrial metabolism by the MT-CB1 and the CB1 signaling is required for memory. So just few take-home message. First, don't smoke weed. This seems obvious for everybody in the room, almost. So the second one is CB1 is a GPCR receptor present at the mitochondrial membrane. This is one of the first identification of a GPCR on mitochondria. And this empty CB1 regulates the oxphos through the phosphorylation. And finally, the empty CB1 links the mitochondrial energetics to the brain function. And to finish, of course, I would like to thank Giovanni, who luckily missed his plane and can be in the room to answer any question. And people from Giovanni's lab, Etienne, who performed all the mito experiment, and Tiffany, and of course a lot of people such as Pedro Gondres, who performed the EM picture. Thank you. Sorry? The N22, yes.
Looks like a mitochondrial per second.
Yes. Yes.
And the protein is outer mitochondrial membrane.
Yep. Yep. But as you see, that there is a very small probability to be in the mitochondria. You have only 40% chance to be a probability of 40% to be to mitochondria. So this leading sequence is not a very strong one. Seems to be not a very strong one. And just
It doesn't work as a leading sequence because we try to put it with GFP alone.
yes, that's true. So it's not a very strong leading sequence. It's required, but not. Yes.
Hi, Oli. So I wonder what is the orientation of this cannabinoid receptor on the mitochondrial membrane. So is it the phase that faces the cytosol on the plasma membrane, it is also facing the cytosol on the mitochondrial membrane, or do you expect the machinery to be in the membrane, in the mitochondrial side?
Always have this question. I think that it is in the same orientation, same topology than the plasma membrane. I mean, we have this the N-terminal, which is facing the cytosol in the plasma membrane. You have the same topology in the mitochondrial membrane.
So it's facing the intermembrane space?
Yes. The N-terminus is facing the intermembrane space. In both cases, you have the N facing the lumen.
So when you localize these alphas and units and so on the inner side of this, there is a paper on G-alpha elongating mitochondria from samples in Barcelona.
We found that actually there is the G-alpha inside mitochondria. We did IP and we perform on isolate mitochondria, and we have G-alpha. We have all the machinery actually.
So yeah, maybe a small question about the bioenergetic defect you see. So if I got it right, you see 30% decrease in respiration, and you link this to a phosphorylation of NDUS2. Okay, complex one. So first question is did you try to check if it was a defect in respiration specific from complex 1 driven electron? So if you use succinate, for instance, would you see no defect in your?
We have no effect on when we measure the succinate dependent respiration. But here we show the phosphorylation status of NWFS2, but actually there is many other proteins that are also phosphorylated or less phosphorylated.
Okay, and
but the complex 2 is not touched.
Okay, so which means that in a way, if you can stabilize or mimic a phosphorylated NDSF2, you would end up having the same phenotype in terms of memory. And did you try this, or would you try to?
We didn't try this.
Yeah. Okay, okay.
On respiration and on memory.
Okay, okay.
Remember, it's
Yeah, okay. Very nice.
Yeah.